Hello, my name is Bilal, and today I'm going to be discussing how I've been using novel light sheet microscopy to study the facial development of genetically normal and genetically mutated mice. Facial defects are amongst the most common congenital birth defects globally, with the most recent data estimating every 0.33 out of 1,000 live births being affected by cleft palate, and every 0.3 out of 1,000 live births being affected by cleft lip. Unfortunately, we don't have a strong understanding for the genetic basis behind facial abnormalities, especially because they arise from the complex interaction of multiple genes as well as environmental factors. In developmental biology, the way we often address this gap in our genetic understanding is by comparing development under normal conditions with development in the presence of a genetic mutation. By comparing the difference in these two scenarios, we can get insight into the function of the gene that was mutated. In the context of studying face shape, we want to be able to quantify and compare the differences in facial shape between our genetically normal and genetically mutated samples. Furthermore, data on cellular dynamics such as cell division and cell death are also very valuable, as these underlying components will ultimately have a major effect on our face shape. In the Hallgrimson lab, we're aiming to study the facial development of regular mice with those that have a mutation in the nitric oxide synthase interacting protein or nocib gene. We chose to analyze nocib because mutations in nocib have been linked to facial abnormalities in the past. Furthermore, mutations in nocib have big implications on cell communication and overall regulation. This is because nocib regulates the amount of nitric oxide levels in our bodies, which is an important signaling molecule. Perhaps more important, however, nocib targets protein phosphatase 2A for destruction. This is really important because protein phosphatase 2A either directly or indirectly is involved in almost all cellular processes. For our analysis of genetically normal and nocib mutant mice, we want to compare their face shape, facial cell division, and facial cell death at different times throughout development. To be able to do this, we need imaging tools that can help us capture developing embryos in both 2D and 3D with high resolution and ideally in a relatively short amount of time. Fortunately for us, light sheet microscopy addresses these needs. Going over the workflow for this project, I first started off by extracting DNA from a sample from each of our mice and analyzed it to determine if the mice were genetically normal or mutated. Now that we know which of our samples are normal and which ones are genetically mutated, we need to prepare them to be imaged in the light sheet microscope. After our samples are extracted, we start off by fixing them, which means we preserve their biological tissue. We then undergo a series of steps to make our samples transparent, and we stain them with antibodies that can detect cell division and cell death. Afterwards, we embed each of our samples in an agarose gel that will allow our samples to be properly imaged and properly positioned in our light sheet microscope. Finally, we can image each of our samples in our light sheet microscope with the optimal laser settings and have each of our samples undergo further processing so that we can get high quality 2D and 3D visualizations of the facial shape, facial cell division, and facial cell death in each of our samples. By following this protocol, I captured high quality visualizations of regular and nocib mutated mice. These photos show a promising result in our ability to capture data that can increase our understanding of facial development. Moving forward, our lab is developing computational tools that will allow us to quantify the cell division and cell death observed in our images. Such data will empower us to relate the differences in cellular dynamics between our normal and mutated mice and directly relate these differences to their final face shapes using diagrams called morphospaces. As you can see in this simple example, a morphospace allows us to visualize all the different face shapes our organism can have based on the changes in a small underlying variable. By using morphospaces, we can directly characterize the unique patterns of cell division and cell death that are associated with normal and abnormal development. Furthermore, we're really interested in diving a little deeper into the signaling mechanisms and overall patterns of gene expression within our normal and genetically mutated mice. Thank you for watching my video. I'd like to quickly acknowledge the members of the Hallgrimson lab for their support and NSERC for funding my work.